So good evening, students. We will be going for the topic protein under biomolecule chapter. This is under your 11th syllabus, and this chapter and this topic is very much important for your NEET as well as your board part of view. So this for the quick revision of the biomolecule or what we will be understanding in this part. We are just quickly recapping on something, that is what is biomolecule. Biomolecules are some chemical substances those are having biological origin. Those are predominantly having carbon and hydrogen, and those are mainly functioning for the building block of our body, that is making different particles or different compounds that is helping us to build our body. This is the biomolecular structure. So biomolecule, under biomolecule there we will be having two parts. First is the micromolecule and second is the macromolecule. Macromolecule, which is the larger part, and micromolecule, which is the smaller part. Okay, so now we will be going for this part, starting this part. Now biomolecule, under biomolecule we will be discussing different parts. Just let me show you, or let me give you a quick recap of this. What are biomolecule? We just discussed what are biomolecules. Just let me clear some doubts of yours. Just this. Yes. Now, what are biomolecules? Biomolecules are molecules which are having this carbon and hydrogen as a predominant form. So, which are having the biological origin, that is biological origin, which are predominantly formed of carbon and hydrogen, and which are the building blocks of our body. Okay, so these are the forms. So under biomolecules we will be discussing four different parts. In case of biomolecule we will be discussing the macro form, which is the polymeric form. We will be discussing what is polymer, what is monomer, and under the this there will be the micro form, which will be the monomeric form. Now what is monomer and what is polymer? Suppose I am just drawing a long chain over here. So this overall long chain, if it is a polymer, then the individual unit is known as the monomeric unit or monomers. So according to this, polymers are macromolecule, and this monomers, the constituting unit which will be making this macromolecule, these are micro parts. Now one by one we will be discussing the macromolecules and their constituting parts. So first we will be discussing the macromolecule, the basic parts, then we will be going for the micro parts. So if we say that the macromolecular part, we will be considering different types. Now students, I will be giving you this clear information that from this chapter many questions will come, and we will be going for the questions as well, what type of questions they ask normally. So just give me the time to give you the basic of this. Then after that we will be going for each and every part. So coming with this, there are two forms. First is the macro biomolecular form, biomolecule, and there we need to understand four parts. First is the protein, okay. Second is carbohydrates, or we can say it as polysaccharides. Third is the lipid molecules, and fourth is the nucleic acid. So we discuss this macromolecules are the polymeric substances or the chain substances, and these are made up of simple monomeric units. So in case of protein, we will be also finding simple monomeric unit, and the constituent macromolecule and the constituent micromolecules for this protein is amino acids, okay. And for polysaccharides or carbohydrate, we will be finding monosaccharides, SACCHARIDE. And for lipid, we will be finding fatty acids plus glycerol. And for nucleic acid, we will be finding nucleotide, okay. So one by one we will be focusing on different parts. So first we will be discussing this protein, this amino acid structures. Then we will be going for stepwise different. And the differences, we need to understand the differences regarding this macromolecule and micromolecule. So if we go for the differences, macromolecule are the larger, or and it is the heavier part, okay. And this is a very light in weight with respect to, as these are the constituent chains, micros are the constituent chain, and these are very light, and these are larger, these are the smallest unit part which are making, these are the smaller part which are making the macroforms. Now their molecular weight is almost more than 800 Dalton, more than 800 Dalton, and here that is less than 800 Dalton. 800 Dalton is the molecular weight calculating formula. We know how to create or how to calculate the molecular weight. If we go for calculating the molecular weight of H2SO4, how would we calculating hydrogen having atomic weight as 1, so 1 into 2, plus sulfur has atomic weight of 32, plus individual oxygen atom will be having atomic weight of 16, so 16 into 4. Now completing, we will be having this 98 gram as our molecular weight, as this is all over, this is a molecule, so molecular weight of sulfuric acid, okay. Now we have completed this differences regarding macroform and microform. Now we will be going for in detail understanding this protein structures. So if we say for the protein part, then we will be going for this. So let me clear this first. Okay, now There's a clear all drawing option. Sorry for that. I forgot this. So going for the protein. So protein is the major form which is making our body. So what is the importance of having this protein structure? Because these are made up of one important atom, which is nitrogen, and this nitrogen gives us the structural compounds or the structural forms of each and every animal, each and every living part. So this proteins are, we discussed that these are polymers, that is the long chain, and it is made up of monomeric unit known as amino acids. Now this amino acids, how we will be characterizing their structure? We now, this is the basic introductory class for all of the biomolecules. One by one we will be going for introduction of different parts. So coming with this, amino acid will be having a general formula of this: carbon bonded with alkyl group 1 bonded with 1 C double bond O OH group and bonded with the H2N group. Now amino acid, why this is amino acid? Because here this particular carbon is bonded with one acid group. This is the acid group and this is the basic group. Why? Because nitrogen has lone pairs, and in case of chemistry we know this, that that atom which will be donating its electron in the form of lone pair, it will be acting as a Lewis base, and that atom or that particular molecule which will be accepting electron, that is Lewis acid. So this is the acid group and this is the basic group. And this carbon, as both acids and basic group are present the same, and this is known as alpha carbon. Now what is this R? This is known as the alkyl group. Now students, you probably know what is alkyl group. In case of hydrocarbons we understood that. In case of alkane, alkane means those are having this CnH2n plus 2 format, that is single bonded aliphatic ring. Aliphatic ring matlab, that is, suppose single aliphatic means the open chain, not the close. Suppose we are forming this carbon chain which are having bonds with hydrogen like this. Carbon has this tendency to create 4 bonds with different atoms. So here it is like this, here it is like this, here it is like this, and again here it is like this. So this is the thing. So here the first carbon at the and the last carbon are do not having direct attachment with each other, so this is known as a open chain. If the first carbon and the last carbon will be having a direct attachment with each other, then this is known as the closed chain. Now in case of open chain, if only single bonds are present, this is known as alkene, and this n will be the number of atom. Here for this, here how much number of atom? 1 2 3 4 5. So here it is C5. Let us calculate the H number. H 2 into 5 plus 2, that is H is 12. So 1 2 3 4 5 6 7 8 9 10 11 12. So it is this. So it is pent n. Okay, and if we remove one hydrogen, it will be getting CnH2n plus 2 minus 1, so we can get that CnH2n plus 1. This is the formulation of a alkyl group or R group. Let us understand that if we go for CH4 methane, if we come with reduction of 1 H, then we will be getting this CH3. Okay, now there is this is the thing, that here will be 3 hydrogen occupied by 3 bonds and 1 bond will be left, which will be giving rise to it this. So from alkene to alkyl group, so here from methane to methyl, so now we will be nomenclating this one by one. So this is the basic class regarding amino acid. We will be further elongating each and every structure, but just remind बच्चों यहाँ पे क्या होता है कि this group, this R group, this H group, here it is C double bond O single bond OH, here it is H single bonded with N, here it is N. What happens, from the acid group one hydrogen is removed in the form of H plus. So what would be happening here, if we elongate this bond, okay, if we suppose try to elongate this bond, now oxygen, it has direct it bond with this one electron of oxygen, one electron of hydrogen. Now hydrogen is removing in the form of H plus without taking its electrons. So what is happening over here, that hydrogen is removed in the form, sorry, hydrogen is removed without not taking its electron, so it is converted into H plus, and oxygen is having hydrogen's electron, so oxygen is converted into having hydrogen electron, and oxygen is converted into this O negative. So it will be having this O negative, and whenever H plus is coming near to this nitrogen, suppose here H plus, now one electron is nitrogen donates one of its electrons to hydrogen, and this electron will also carry one negative charges. So negative positive, that is here this negative and positive, this positive charge and this negative charge will oppose each other, so hydrogen will be neutral with having one electron. And also, so as this will be opposing each other, we will be not putting any charge on him, on this hydrogen, and nitrogen, by losing its one electron, it will be here, it will be having only one, but it lost its own electron, it will be having this positive charge. Now this electron will be bonded with nitrogen, and there this NH3 plus will be there. So what finally forms from this part, that is we are having carbon, R group, C double bond O, O minus, here H3N plus, and here this hydrogen. So one molecule having negatively charged and also positively charged is known as Zwitter ion, and this ion will not be following any electrophoresis, Phoresis, electrophoresis. So electrophoresis means the movement of particular ion. So this part is the anionic part and this part is the cationic part. So they will not be moving to any toward any electricity, so or electric, they will not be moving toward any electrode. So this is known as, this part is known as electrophoresis. So as they are in Zwitter ion, they will not be going for any electrophoresis. and the particular pH in which this zwitter ion is formed is known as the pi value. So there are a lot of questions regarding zwitter ions pi values, and there are a lot of questions for I will be separately making an video regarding net, the CSIR UGC net, and also for the NEET UG syllabus purposes, what is zwitter ion, their applications, their different forms, etc, etc, and also the how the peptide bonds are created. Okay, now this amino acid, suppose this is important. So suppose this one is the amino acid which are having this C double bond OOH and having this H2N in normal condition. This is the first amino acid. Suppose the second amino acid is HNH. This is second, this is this, and C double bond OOH. This is second. There will be third, there will be fourth, etc, etc. One second. There will be third, there will be fourth, etc. So this, from here, removal of one water molecule will be giving this bond, this creation of carbon and nitrogen bond, which is known as peptide bond. And in case of us, in the inside the cell, through the translation process, when mRNA molecules with the help of ribosome, it is forming the protein or going for the translation process, then peptidyl transferase enzyme, this is very important, transferase enzyme, it is helping the creation of this peptide bonds. And whenever like this 1 2 3 4 like this, whenever more than 100 amino acids are conjoined together with respect to peptide bond, they will be forming the protein structure. We will be in detail going for the primary structure, secondary structure, tertiary structure. It is important for mRNA with ribosome, and obviously there are tRNAs which are grabbing the amino acid from the cytoplasmic amino acid pool. These are joining one by one amino acid with respect to the codon sequences, and they are creating this particular protein. So we will be in detail discussing all of this, but as this is the basic of biomolecule, we will be just discussing the basics. In next part we will be going for basic of carbohydrate, basic of DNA, that is the nucleic acid, basic of lipids as well. Okay, so this is the first part of biomolecule basic. We will be going for this second and third, basic of biomolecule 1, basic of biomolecule 2, the basic of 1 series, the basic 2 series, second series, and the basic 3 third series. That you will be having this knowledge to what to read for, questions are coming from where. Then we will be elaborating the proteins. Then there will be coming this series proteins, there will be coming this series lipids, there will be coming this series nucleic acid, and here there will be DNAs, majorly DNAs and RNAs as well, and this carbohydrate. Now we will be having total 1 2 3 4 5 6 7 and one important enzyme, total 8 videos with respect to this part. Okay, we completed the basic of this. Now we will be completing basic 2 with respect of, first there will be carbohydrate, basic, plus we will be completing the lipid basic. Okay, and here we will be completing nucleic acid basic part, introductory part or something. With nucleic acid के साथ we will be थोड़ा बहुत we will be discussing what is enzyme. Now the actual part with respect to question answer, it will be coming to this series. This, go for this. Okay, there will be separate playlist for this and this and this and this and this. Okay, go for this. Thank you for watching this video, and it is a humble request to each and every students, please do subscribe, and if you think that this video is very much informative for you, then please go for a like as well. Thank you. Have a good day.